You know, when you take a pill, you trust it's going to work. But how can we actually be sure? Behind that simple little pill is a huge scientific challenge, proving that every single one performs exactly the same. We're about to dive into a hidden problem in the pharmaceutical world, and the solution? Well, it's surprisingly elegant, and it was found in a program you probably have open on your computer right now.
So let's start with that big question. How do you prove that two pills are identical? You might think, well, if they have the same active ingredient, they're the same, right? But it's not that simple. Not at all. The real test is how that ingredient gets released into your body over time. A generic drug has to perfectly mimic the brand name version, and trust me, that is a really complex puzzle to solve.
To really get a handle on how scientists tackle this, we need to look at something you could call the pill's performance fingerprint. It's this unique signature that's the key to proving a pill does exactly what it's supposed to do.
And that fingerprint, it's called drug dissolution. Basically, it's a measure of how much of a drug dissolves at different points in time. You plot that data and you get a curve. That's the unique signature for that pill. So if two pills have the same curve, they're considered bioequivalent. That just means they'll work the same way in your body. It sounds pretty straightforward, right? But comparing those curves, oh boy, that's where the real trouble began.
And this brings us right to the heart of the dilemma, the big headache that was plaguing pharmaceutical scientists for years. They knew exactly what they needed to measure, but the process of actually doing it, well, it was a total logistical nightmare.
I mean, listen to this quote from the very researchers who ended up solving the problem. It says it all. The analysis was done by hand. Just let that sink in for a second. Critical data for drug safety was being crunched with calculators and spreadsheets. It was incredibly slow, and as they point out, transcription errors are a common problem. Think about that. A single misplaced decimal point could derail an entire drug trial.
So what did by hand actually look like? Well, it meant wrestling with these super complex nonlinear equations, plugging in every single parameter one by one, and just spending hours and hours on calculations. The alternative? You had to shell out for really expensive professional software that wasn't even designed for this specific task and still made you define every equation manually. It was slow, it was expensive, and it was just dangerously prone to error. But then, in 2010, a team of researchers from China Pharmaceutical University came up with a brilliant solution. And it came from a very, very unexpected place. Just take a look at the timeline here. The math, the actual theories for comparing these drug profiles, that was all worked out back in the 1990s. But then, look at that gap. For over a decade, there was no practical, easy-to-use tool for scientists to actually apply that math. The theory was there, but the bridge to get it into the everyday lab work was just missing. Until 2010 and the arrival of DDSolver. And here it is. The solution wasn't some fancy, standalone, complex new program. Nope. It was a simple, free add-in from Microsoft Excel. Instead of forcing scientists to learn a whole new system, the developers brought the solution right into the environment they already knew and used every single day. I mean, what an absolute stroke of genius.
So why was Excel so perfect? Well, think about it. It just checked all the right boxes. It was easy to use, pretty much every scientist on the planet already had it, and it was flexible enough to handle all the complex calculations. This one choice basically guaranteed that the tool would be adopted quickly and widely, and it completely democratized this crucial step in drug development.
So what could this humble little add-in actually do? Let's take a look under the hood and see the powerful scientific engine they built right inside a simple spreadsheet. Get this, 40. That's the number of different, complex mathematical models for drug release that were built directly into DDSolver's library. This gave scientists an incredible amount of power and flexibility to analyze their data without ever having to manually code or define a single equation. It was all just ready to go. And here's the really crucial part. DDSolver automated the exact statistical methods that regulators, like the FDA, actually use. Take a look at the similarity factor, F2, on this chart. That right there, that's a key metric for proving two drugs are bioequivalent. Before DDSolver, calculating it was this tedious manual nightmare. Afterwards, it was literally just a click away. This slide just illustrates the new workflow perfectly. On the left, you've got the raw data, just columns of time and percentages. A scientist would just plug that in, and boom, DDSolver would instantly generate the output you see on the right, all the complex comparison factors calculated perfectly. It turned a laborious, error-prone task into a simple, streamlined process.
So what was the real-world impact of all this? What happened when this powerful, free tool was put into the hands of scientists everywhere?
The impact was massive, and it basically came down to three things. 1, it made really sophisticated data modeling accessible to everyone. 2, it simplified the complicated regulated process of comparing drug profiles. And 3, this is the most important one, it drastically reduced the risk of human error in a field where accuracy is everything. And that means better data, which of course leads to better drugs.
And that's really the ultimate takeaway here. DDSolver wasn't just another program. It was the first of its kind, designed for this one crucial task. And because its creators made it completely free and available to everyone, it didn't just advance their own research, it pushed the entire field of pharmaceutical development forward.
You know, the story of DDSolver is such a great reminder that innovation isn't always about inventing something brand new from scratch. Sometimes it's about seeing the hidden potential in the tools we already use every single day, which really makes you wonder, doesn't it? What common program sitting on your computer right now could be adapted to solve the next great scientific challenge?